Am I audible and visible, guys? Okay. So let's get started. Last class we discussed the conventional methods to model the diseases, which is the animal models and computational modeling. And we also discussed about our organoids and how they help in disease modeling and drug testing. And then we finally moved on to 3D printing and 3D bioprinting as well. And we saw a live demonstration as well. In today's session, we'll be discussing about the success stories of lab grown tissues and organs. And then we'll be moving on to the challenges which we face while creating uh, a lab grown organ. And then finally, we'll be concluding this module by discussing the ethical considerations revolving around lab grown organs. So, let me start the presentation. Is my screen visible to you guys? Give me a thumbs up. Okay. So, let's
Yes,
move
it's
on.
visible.
Okay. That's nice. So, let us discuss the success stories. While replicating a fully functional organ remains a challenge, significant progress has been made in tissue engineering, offering promising solutions for various medical needs. And the first success story which we are going to discuss today is about skin grafts. Lab grown skin grafts are already a reality for us. They are a major advancement in treating burns, chronic wounds, and even some skin diseases as well. So, what is the process involved in it? Doctors can take a small sample of healthy skin cells from a patient and grow them in a lab setting on a specialized scaffold. We have discussed about scaffolds earlier. So, once enough cells are produced, they are transplanted onto the wound site. So, what is the process for taking skin sample from a patient? Do you guys know about it?
A biopsy.
Yes, you're right. Uh, so if you can't hear it, you can refresh the page. So let us discuss about the process for taking the skin sample. The first thing is taking the preparation for taking the biopsy, which is the area chosen for the biopsy, usually the upper arm, will be cleaned with an antiseptic solution to minimize the infection risk. And local anesthesia might be given to numb that area. The second one is taking the sample. We use a tool called as a punch biopsy, and that extracts a millimeter sized circular piece of the skin. So this sample is typically less than 4 millimeters in diameter, and pressure might be applied to stop any bleeding after taking the sample. Now bandage is applied, and then the skin sample is transferred to a sterile container and transported to a specialized lab for iPSC generation. So this is the process. Now, what are the advantages of the lab grown skin grafts? Since the cells come from the patient themselves, the risk of rejection is minimized. And lab grown organs can also promote faster healing and can reduce the scarring compared to the traditional skin grafts. Now, what are the traditional skin graft procedure? It's a surgical procedure that involves transplanting a healthy skin from one area of the patient's body, which is also known as the donor site, to the affected area of the same patient's body, which is the recipient site. So, this lab grown skin grafts can also help to improve the appearance as it closely resembles to the patient's natural skin tone and texture. So, I hope you guys got this. If yes, give me a thumbs up. Yes. So, let us now move on to the next success story, which is about cartilage and bone repair. So, traditionally, cartilage and bone injuries were treated with limited options like grafts from other parts of the body or from the donor tissue. These methods have drawbacks like limited availability or potential complications. Now, scientists have successfully engineered cartilage and bone tissues for transplantation. So, this involves the combination of cells, scaffolds, and growth factors, which we have already discussed in previous sessions. So, let me give you a gist of it. So, cells, we use these stem cells or specialized cells such as chondrocytes, which are the cartilage cells, or osteoblasts, which is the bone cells, to uh, create the lab grown tissue. And we use scaffolds as well, which is the biocompatible material used to create 3D structure to mimic the natural bone or cartilage, and it supports the cell growth as well. So, what about the growth factors? They are signaling molecules that are added to stimulate the cell proliferation and cell differentiation. So, do you guys know the difference between proliferation and differentiation?
I think differentiation is like the increase and growth of cells, and proliferation is like the spreading of them.
Uh, I think you switch the roles of both. So, let me explain it to you. Proliferation is nothing but cell division, like the cell makes a copy of itself. Yeah, multiplication of cells. Now, differentiation is a process by which a single cell transforms into various specialized cell types with unique function. Yeah, Gorija and Jyotin, you guys are right. So this is the property of stem cells, right? We discussed in the previous sessions as well. So what are the applications of the cartilage and bone, which we made in lab? So these can be used to repair damaged joints in knees and shoulders and in other areas as well. And bone grafts can be used for facial reconstruction after accidents or surgeries, and potential for spinal fusions and other bone repair procedures as well. So the third one is biovessel engineering. So because creating bio functional biovessels is a very crucial step towards engineering complex organs, right? And these vessels are very much essential for transporting oxygen, nutrients, and waste products throughout the tissue. Now, what is the challenge in it? Developing blood vessels require not just building tubes, but also we need to ensure that they are lined with appropriate cells, such as the endothelial cells, uh, and have the proper structure to withstand the blood flow as well. So, what is the technique used here? Uh, the first one is bioprinting, which we have discussed. This technology allows for precise printing of 3D structures, including the delicate network of blood vessels, but this is really difficult to print the uh, blood vessels as well. The second technique is called as cell seeding, where we encourage the growth of endothelial cells on scaffolds to create functional vessel walls. The third process is decellularization of an organ, which again we discussed earlier. This involves removing the cells from a donor blood vessel, leaving behind a natural scaffold that can be repopulated with the patient's own cell. Now, how do we check whether that particular organ is completely uh, decellularized or not? What is the process? Anyone in the chat? So, what is the process which we use to check whether a particular organ is completely decellularized or not? Do you guys have any idea on that? Or should I explain now?
I think you can give us a hint.
Uh, so this is something related to DNA. Can you guess?
So you check the genes of the patient.
No, here we are not talking about the patient. So last class I showed you a decellularized heart rate. So how with that heart sample, how do we check that whether we have removed each and every cell from that heart or not? Basically, we should be left with only the extracellular matrix. How do we check the purity of that organ? Yes, Gareja is right. So let me explain. So you have learned about PCR in our in session, right? So what we do is we take a small sample from that organ and we use PCR technique in that. So if we find high amount of DNA present in that, the sample is not very pure. Why? Because DNA is present in the cells only, right? So if more cells are present, then obviously more DNA will be present. So the less the DNA which we get in our PCR sample, the more the more purified the organs. Did you guys get it? Okay. Okay. Now let's discuss about kidney organoids as well. So while a fully functional and transplantable kidney remains a future goal, scientists have made significant progress with kidney organoids as well. Uh, so where these mini kidneys are valuable for us in drug discovery. where we study how drugs affect the kidney function in a more realistic setting than traditional cell cultures. We use these kidney organoids in disease modeling as well. So, where we understand the mechanism of kidney diseases, where we understand the mechanism of kidney diseases and we develop potential therapies for it as well. Uh, these are the building blocks for future bioengineering as well. So, the knowledge which we gain from kidney organoids can pave the way for creating more complex and functional kidneys in the future. Do you guys have any doubts so far, or can we move on to the next topic for the day? Yes, give me a thumbs up. No doubts. Okay. Um, So the next topic is, what are the challenges we face while replicating a functional organ in a lab? The first one is the complexity of the organs. So the analogy is like uh, the workers analogy, which we discussed in the last session as well. So there are, let's say, for creating a building, there are different workers, right? Specific workers specialize in laying the bricks, and some are great electricians, but their overall job is to create a building. So, similar to this, organs have many different types of cells working together, like the muscle cells in the heart or the nerve cells in the brain. So, recreating this mixture and ensuring that they cooperate well is very crucial here. The second challenge is creating the complex transportation system, which is also known as the vascularization. So, just like the workers needs roads and pipelines to deliver goods, organs rely on a network of blood vessels, which are also known as the vascular network, to carry oxygen and nutrients to all these cells in the body. So, building this intricate network within a lab grown organ is a major hurdle for us. So, what is the third challenge? It is the cell communication and organization. So, cells within an organ don't work in isolation, but they work as a team. They need to send and receive signals to coordinate their actions. So, understanding and mimicking these communication pathways is very much essential for a lab grown organ to function properly. Now, what is the fourth challenge? It is the long term sustainability. What is that? Lab grown organs need to be able to repair themselves and grow and adapt like natural organs do. Natural organs are composed of various cell types that works in harmony, right? And these cell types are organized in specific structures and communicate with each other through complex signaling pathways. While the lab grown organs can replicate some of this complexity, it may not perfectly recreate the intricate cellular microenvironment and interactions present in natural organs. So, this can be a limitation for their ability to respond to damage and initiate a repair process. Now, what is the fifth hurdle? That is the immune system hurdles. So, even if we use a patient's own cell, the body's immune system might still see it as a lab grown organ and reject it. So, why? Do you guys know the reason for it? Any guesses? why our immune system can still see a lab grown organ as foreign. Uh, but how come it is a gene difference? We are basically using the patient's own cells, right? We have discussed about this earlier. Try to recall the previous classes. What is the what is an important thing used to create a lab grown organ? What is the base?
Bioink.
Yes, yes. Scaffolds. Bioink. Uh, Bioink mainly consists of the stem cells, which is the cells of the patient, right? So it is. It doesn't cause much of the abnormalities, but major problem is with the scaffold or the biomaterials which we use. So because of that, our body can still see it as a foreign object, which can lead to rejection. Now, there is one more challenge, which is called as the epigenetic memory. So, let's say we took a skin cell and we transformed it into iPSCs, and then from that, we created a nerve cell. But we cannot say that this nerve cell will completely behave like a nerve cell only. This might have some memory of its past and it might start behaving like a skin cell. It might start behaving as a skin cell also, and that is also a challenge which we face. And this is not called as epigenetic memory. And you'll be discussing more about this in the next week, which is which Vaishnavi will be teaching to you guys. So if the scaffold is also taken from the patient, can we resolve this immunity problem? Uh, yes, most probably yes. But still, there are other factors, right? Everything cannot be completely derived from a patient. We use artificial growth factors as well. So you get it? Okay. And iPSCs can have chromosomal abnormalities as well. Because while transforming a mature cell back into a pluripotent state is a very complex process. And this process can put a significant stress on the cell's machinery responsible for DNA replication and cell division. So, under the stress, errors can occur, which can lead to chromosomal abnormalities in the iPSCs. So, and these the special factors which we talked about, right? The Yamanaka factors, they also play a role in causing the abnormalities. So these were the challenges which we uh, see or face while replicating functional organs in a lab. So, do you guys have any doubts so far? Can we move on to our next topic? Yes, I see very few responses, guys. What about others? What are you guys doing? Give me some thumbs up, guys. That's a good response. So, before moving on to the ethical considerations, let us invite Diksha to show the exact model of trachea which he made in Blender. So, let's welcome her.
Okay, so let me share my screen. Uh, I hope you guys can see my screen. So this model was made on Blender. So as I mentioned earlier, it is hard for us to model like irregularities in Fusion, but it is possible in Blender. So this is what I modeled on Blender. And another thing I wanted to tell is why 3D printing and 3D bioprinting are not the same things. They are different from each other. So 3D printing is basically we print like solid materials, but bioprinting this is designed to print like liquid or gel. So it should be 3D bioprinters are more sensitive. They are designed to handle more sensitive materials, and also we are dealing with live cells. So it is harder to print because we have to ensure biocompatibility. So that is why a 3D bioprinting is harder compared to 3D printing, and still people are working on it to make it more efficient. So yeah, this is the model which I made. So this is the larynx part. Then we have our trachea, and we have the rings of cartilage, and I made a primary bronchi. So I'll just show you like how I made it in basic. So let me just hide this. Okay. So basically, in Blender, one of the major things is adding an object. So I'll show you how you add an object. So this is going to be your workspace. And to add an object, you're supposed to go to add here, or you can use the shortcut key Shift A. And here under mesh, you have many objects you can add, or you can add other things like text, etc. So I basically added a cylinder first, something like this. Then you can scale the object as well. So here under transform, here you have your scale. You can increase your width and breadth and height also. So that is scale. So this is how it look. Then to make irregularities, we'll have to go to either our edit mode or sculpt mode. So currently we are in object mode. So we can go to edit mode, and before adding irregularities, you will need something called as loop cut. So you'll have to cut your surface into many parts. To do that, you can just use the shortcut Control R and increase the number of cuts using your middle scroll mouse button. So once you're satisfied, just click enter and enter. So this is you'll get your loop cut. And so here there are three options next to edit mode. One is vertices select, one is edge select, and one is face select. So you will have to turn on something called as proportional editing. So that is an option here. So if you turn that on, so you can basically select some vertices like this and just move it. So now you can see that you can move it as you want. That will help you to add some irregularity. So that is how you move it, something like this. So that is one method of adding irregularity by moving the vertices. So first we added a cylinder, then we cut the cylinder into parts, and then we move the vertices according to our needs. So that is one way of adding irregularity to any object. Other way is, instead of using edit mode, you go to sculpt mode and you have an option called as inflate. So using your inflate option, you can add deformalities. So you just here you have a strength of the brush. So this is your blue color is your brush, and this is you can increase the radius of the brush, and you can increase the size or decrease according to your need. And once that is set, you can just move it along the cylinder, so you can see the shape is changing a little. So you can rotate it, move it. So to rotate it, you just use your mouse, middle mouse button, and rotate across the workspace. Or you can use your something like in Fusion on the top right hand side, you have a XYZ axis. You can use that to move. So there are other options also like crease, smooth, flatten, fill, etc. So using these various options, you can uh, add irregularity and deform the object a little. So here you have something called as grab. So grab basically helps you to move a vertex a little. So if you see now, you can move it around. So I advise you guys to explore these options. So it's actually easier if you want to model it on Blender, but only thing is you'll need to get a hang of all the tools and yeah, that so something like this. So that's how you add a irregularity to the object. So once you're happy with what you've done, just come back to object mode. So you can add other objects also. So you can add like cube, circle, etc. So for now, since we are modeling the trachea, cylinder is enough. So to add the primary bronchi also, you will have to add like more cylinders and move it. So to move an object up or down, you use your grab button, which is a shortcut key is G, and then select the axis in which you want to move it. So let's say I want to move it along the Z axis. Just after clicking on G, just click on Z. So that will you can move it down. And in case you want to rotate it, just use the shortcut R. R and select the axis. So let's see if I want to rotate about my Y axis. Click Y. You can rotate it. And again, if you want to move it, you can move it. So if you click, if I say once I rotate it, my object has tilted, right? Now, if I want to move it along that direction and not the original XYZ axis, I'll have to click on G, and instead of pressing Z once, I'll have to click Z twice. So that will come to my local Z axis, which is this axis. Then I can scale an object also. The other shortcut key for scaling an object is S. S, and again press any axis on along which you want to scale it. Or in general, if you click scale, it will scale equally along all axes. Or you can go to transform and scale it here. So that is one way. You can also mirror an object. So there are many modifiers present in Blender which make your work simple, like mirror, array, smoothen. So if I let's say I'm happy with the cylinder and I want to smoothen it, just select the cylinder, right click on your mouse, and you have a shade smooth option. So you can do that. So that will smoothen your object. So same way you can add irregularities to the cylinder also. Go to your edit mode first, but before adding that, you'll have to add loop cut. So use Control R, cut the thing into many parts, then go to sculpt mode. You can use any option as I said before. You can use inflate and So just move your brush along the side, or you can add a flatten a little if you want to push any part in, something like this. Or you can grab and move. So so again, once you are happy with what you've done, just go back to object mode, then right click shade smooth. So that's how you move it. Now, if I want to join two bodies, so these two are two separate bodies. So if you see in your left right hand side here, you have this have a set of objects. So currently I have hidden other objects and only these two objects are open. So if let's say I want to combine these two, just select the two objects. So to select both the objects, you'll have to use shift. So select one object, click on shift and select the other object. Then you can go to uh, either you just use Control J or right click and click on join. So that will join your object. Now both are a single body. So that is one thing. So that is how I uh, added irregularities to the trachea part, which is not possible in Fusion. So, yeah. So let's add another cylinder. So you can again go to your sculpt mode, but yeah, you'll have to add loop cuts and just sculpt it like according to your need. So again, once you're happy, just go back to your edit mode, right click, shade smooth. Then you can combine the bodies using Control J. Uh, Once this is done, so this is the basic trachea part. So you can add more detail. Like if you spend more time on this, you can add more detail. So I just showed you how you can add irregularities. So same way you can add the rings of cartilage also. You can add some small cylinders. So go to your add cylinder, make it smaller, and you can scale it along both your X and Y. You can move it up a little. So something like this. And again, you can add irregularities to this. So let's say you want to duplicate an object. You can either use the mirror modifier or you can use a simple shortcut key called Shift D. So if I select my body and click on Shift D, it basically duplicates my object. And then I can move my object anywhere. So let's say I want to move it along my Z axis. So do G and Z. and you can move it along Z. So something like this. So again, you can go to your sculpt mode, I mean, go to edit mode, add in loop cuts, then go to your sculpt mode and move it a little, make it a little irregular. Then shade smooth. So you can do it like that. Uh, so you can add in more rings, you can add in your creativity. So that is about rings of cartilage. Now let me show you how you can add in color. So on your right hand side, you have many options here. So the second last option which says material, click on that. So with your body selected, click on new, and here under base color you can change your color to whatever you want. So if you want a little orange shape, you can, then you can add in, you don't want it to be metallic or you want it to be rough, you can add in your creativity here, make it a little lighter, something like that. So same way you can do it for the rings of cartilage also in case you want any color. Any doubts till now? You can unmute and ask. Okay, if there's no doubts, then so this is
How
basically
did
how I,
you make the larynx?
yeah, I'll come to that part now. So larynx, I added, go to your add option, mesh. You have a circle option. So, so if you click on circle option, this is how you get a circle. Move it up like here and move it a little towards your center here. So now you have the circle, but you want it to be larynx shape, right? So what we do, go to your, from object mode, go to your edit mode, and then you can extrude your vertices. So to extrude your vertices, you'll just either you can go to vertex and extrude vertex or click on E option. E, so you can extrude. But you want it along a particular axis. So let's say I want it along Z axis. I can do my Z, move it up a little, something like this. Then what I can do, I can go back to my object mode, then right click shade smooth, and I can, since this is only a plain surface and I want a little thickness, what I can do, I can click on the object. So here you have, again, I showed you materials, right? Same way here, there's a modifiers option here. So you go there, search for solidify. So if you give a solidify modifier, basically adds a little thickness to your plane. So for now, this is enough. So once you're happy with the thickness, you can just, there's a arrow here, just click on that arrow and click apply. So when you go back to your edit mode, this is how it will look. So now what I can do is same. I can click on any two vertices with my proportional editing turned on. I can move it, something like this. So I can change the scale of my proportional editing. Like I can make the circle bigger so the influence will increase, or I can decrease it using my middle mouse button again. So if I want to move it along my x-axis, something like this, get it like that. So if you see, Now you can see a little. So this is how I added the larynx part. So same way you can do for like let's say I want to deform this a little down. Then click on the vertex GZ, move it down. You want to increase your influence, just increase this. So you can see this. So same way you can do it on the other side also. Just click on your vertices. So as I told you before, if you want to select multiple vertices, you use your shift option. Shift and select the vertices. Then move it up or along any axis which you want. So you get something like this. So you can move this part also. You can select the bottom vertices. Move it up. So you can add in, or next you can go to your sculpt mode and grab any vertex and move it up or down as you want. So here, so this yellow color, there's a dot, right? So that is basically showing your vertex. So left click on your mouse and move it. That's how you can move it. Something like this. So, so yeah, as I said earlier, in Blender there is no timeline, so you can't see all the steps which you did till now, but you can always use your undo and redo option. They work. No, redo won't doesn't work. Only undo works. So if you're doing anything, you'll have to be very careful. So that is how you add in irregularities. Is it clear till now? Any doubts you can
Yeah,
ask.
guys. Mm.
Okay. So again, you can change the material here. So you can select this and select on material and give it a little darker shade if you want. So that is how you add. You can add color. Now, so this is, there are different modes. So if you see on the top right hand side, you have your viewport shading. So this shows you how your object looks without any color. And in next mode is your shading mode, but with the shade on. So here you can, here it will show you the colors you have applied. And then we have a render mode. So in render mode also there are many properties. So here you have your render property, output, etc. So here you can choose different cycle. So this is more helpful when you're using animation or you're adding like animation. So rendering works well that time. So there are many options like your output, how, so there's an option called render here also. So when you render an image, how you want your image to look, that will be decided in your render view. You can change your resolution of your output. You can change the frames, but that is not needed for now. You can give different color background. So now currently this is sky texture, right? This is how it looks. Okay, so let me hide this and let me unhide this. I think so yeah, you can see this how it looks. For now, let me hide. Delete this. Delete this also. Yeah. So this is how it looks. You can add in a camera. So go to add, go to camera. You can add in your camera. You can shift. So here you have few views here. So you can go to your camera view. So this is how your rendered image. This is the view in which you're going to get your final image. You can move your camera accordingly. You can add in some extra light. So how you add that? Go to add, go to light, and you can add in different lights. So currently I've added a area light here. This is your area light just for more brightness. You can increase the brightness of your light also. So, so if you are satisfied with your image, then what you do, you go to render and click on render image. I mean, if you're satisfied with the view and your render properties are fine. So here I've given my power for the light as I can increase, decrease the power. So that will affect your final rendered image. So once I'm happy, just click on render and render image. So this is how you will get your image. This is your final output basically. So this is some basics of Blender. I hope you guys understood. So I'll recommend you guys to like use the software by yourself, try out different options, and yeah, once you get used to it, you can try if you want. You can model your trachea again here. Any doubts till now? I think I'm done with my part. Okay, if no doubts, then Samiksha can carry on now.
Okay, that was awesome, right, guys? So you learned something new, how to use new apps, how to use new softwares, and I think now we can move on to the ethical considerations. Shall we? Okay, guys. So we saw how iPSCs are created, but how do we get embryonic stem cells, guys? Is the embryo killed? No. Then how do we get it? Any ideas? ICM, inner cell mass. Yeah, that's right. But from where do we get that ICM? It is present in an embryo, right? Extracting the cells from where? From an embryo? So let us discuss about this in detail. So what are the ethical considerations revolving around the embryonic stem cells, which can be solved using iPSCs. So let us discuss about these questions in detail. Uh, that we'll discuss about that, Parija. So acquiring embryonic stem cells is a very complex process with ethical considerations. So first, let us discuss how embryonic stem cells are uh, obtained. So the first process is from donated embryos from in vitro fertilization. How many of you know what is IVF? Okay, many of you guys know about it. So let us discuss about it in brief. So in the lab, the retrieved eggs and the sperms are combined. So here there are mainly two fertilization methods. The first one is conventional insemination, where the sperm and the egg are mixed in a dish, allowing fertilization to. Okay, no need to worry about it, Korija. So, what happens in conventional insemination? The sperms and the eggs are mixed in a dish, allowing fertilization to happen naturally. The second process is intracytoplasmic sperm injection, which is also called as the ICSI process, where a single sperm is directly injected into the cytoplasm of an egg cell. So, this increases the fertilization rate for certain cases. Now, the fertilized eggs, which are also called as the embryo, are monitored for cell division and development over the next few days in a controlled environment. After few days, usually 3 to 5 days later, one or more healthy embryos are chosen to transfer into the woman's uterus. Now, the source for the embryo for extracting ESCs is IVF procedures. We use the embryos which are no longer needed for implantation. So, what is the process for it? With the informed consent from the donors, the embryos are donated to research institutions. Now, what is the target stage of the embryo? The target stage is the blastocyst stage. So, blastocyst is nothing but a ball of cells typically formed after 4 to 7 days of fertilization. Why is blastocyst a target stage? Because it contains something called as the inner cell mass, which is the source of ESCs. You can see in this pic as well. This is the fertilized egg. We move on to the two-celled, four-celled, and then the eight-cell stage. Then we move on to the 16 cells, I think it's 16 celled only, which is the morula, and then comes the blastula, and then we finally move on to the blastocyst stage, which looks like this. So you can see some cells here, right, inside the blastocyst. This is nothing but the inner cell mass. Now, scientists carefully isolate the ICM from the blastocyst, and then these isolated ICM cells are then cultured in specific laboratory environment to grow and maintain their stem cell properties. So this is the first method to obtain ESCs. Now let us discuss about the second method, which is the somatic cell nuclear transfer, also known as the SCNT process. So what is the source for it? We use the patient's own adult cell, for example, a skin cell, and a donated egg. Now, what is the process? The nucleus which is the genetic material, is removed from the donated egg cell. And then the nucleus from the patient's adult cell is inserted into the enucleated egg cell. Enucleated means the one which doesn't have the nucleus. Now, the modified egg is stimulated to develop into an embryo. Now, this is similar to IVF methods only. After we created an embryo, and then we wait for the embryo to get into, wait a minute guys. Now we wait till it becomes into a blastocyst stage, and then the blastocyst is isolated, and then the ICM present in it is used to derive the ESCs. Now, there is a famous example for this process. Do you guys know what it is? It's a very famous one when it comes to genetic engineering. Any ideas, guys? Any ideas? Or can I explain? Okay. So, there is a famous example for this somatic cell nuclear transfer process. Do you guys know what that example is? No. Like you have even read about this thing in your textbooks. Yeah, the sheep. Which sheep? What is the name for it? Rosie is a cow. Dolly. Yes. Yes, yes. Dolly the sheep is a very famous example for gene cloning. And how did they create Dolly? So let us see how did they create. So there were three sheep involved in it. The first one is Finn Dorset sheep, from where the nucleus was taken from its adult cell. Now, the second sheep is the Scottish blackface sheep, from where the egg cell was taken and its nucleus was removed. Now, what did they do is they put this nucleus from the Finn Dorset sheep into the egg cell. Now, they grew this embryo in lab till it till like blastocyst stage or for 4 5 days, and then they transferred this embryo into a surrogate mother sheep, which is also a blackface sheep. Now, what is uh what is the outcome of it? Like, how did what is the genetic similarities of the baby sheep with the parent sheep? Since we removed the nucleus from the egg cell, it doesn't have any genetic material, right? Because only the nucleus contains the genetic material for a particular organism. We completely replace that nucleus the with the Finn Dorset sheep's nucleus. So Dolly exactly has the uh, Dolly has the genetic makeup which is exactly same as the Finn Dorset sheep. So this is where the cloning was successful. Isn't it amazing, guys? Yeah. So do you guys have any doubts so far? Did you guys understand the concepts which we have discussed till now? I see many thumbs up here. Okay, that's great. Now, let us finally move on to the ethical considerations part of it. Now, the first one is the source of stem cells. These stem cells are derived from an early stage of embryos, which have the potential to develop into any cell type in the body. However, their use raises ethical concerns for some people who believe that it destroys a potential human life. So it's correct only, right, guys? If we have implanted that embryo in a surrogate mother, that would have become a human. So this raises an ethical concern. And what is the alternative source for it? As we have discussed earlier, it is nothing but our iPSCs. And scientists are actively researching the alternative sources of stem cells, such as iPSCs. And this offers an ethically less controversial approach. The second concern is about designer organs. The ability to engineer organs could lead to the creation of designer organs with enhanced traits, such as increased resistance to a particular disease or extended lifespan. This raises questions about the fairness and potential misuse of it. So, the what is the equality of access? Even if it was allowed to create designer organs, would designer organs become a luxury available only to the wealth, which in turn increases the existing inequalities present in healthcare access? So, there is a need to ensure equitable access to this life saving technology. The third one is nothing but resource allocation. So, let's imagine you are having a pizza party with your friends, but there are only four slices of pizza left. Resource allocation is like figuring out how to fairly share those four slices amongst everyone. First, we need to identify the need. We'll figure out who needs the resource. Uh, in the example of pizza party, for example, everyone would want a slice, right? But we need to prioritize it. Not everyone might have the same need. Maybe one of your friend hasn't eaten all day and you have just completed your lunch or dinner. So they might need a bigger slice of pizza or they'll have to be given a higher priority. Now the third process is distribution. Once we identify who needs what, we can distribute the resources fairly. For example, in a pizza party, you might cut the slices equally, which is equal allocation, or we can give a bigger slice to our extra hungry friend. So, this analogy works exactly the same way when it comes to organ donation or lab grown organs as well. Uh, Currently, the demand for donor organs far outstrips the supply, right? But with lab grown organs, the potential for a limitless supply exists. But who gets the access to these organs when they become available? A clear and fair criteria for allocating lab grown organs will be crucial. Factors like medical urgency, compatibility, and a patient's overall health could be considered. So, this is all about resource allocation. Now, let us move to the last ethical concern, which we'll be discussing today, which is the use of animals. As we have discussed in our first session, Certain aspects of lab grown organs research might involve using animal models for testing and development. This raises ethical concerns about animal welfare as well, as new materials and techniques used in lab grown organs might be tested on animals to study the biocompatibility, which is we test how well the material interacts with the living tissue. This ensures the safety of the materials for eventual human use. Now, researchers should strive to minimize animal usage by employing alternative methods wherever possible, such as computational modeling or advanced cell cultures. So, this is all about ethical considerations, guys. Did you guys get it? Did you guys enjoy it? So, let us move to our activity. How to make transplants more accessible, guys. As we have discussed in the first session, the current methods for solving the problem of organ failure is traditional organ transplantation only, because lab grown organs are might be a possibility in future, but in present we are still researching on it, and in present the donated organs are the only solutions. Now, with this, how to make transplants more accessible, how to find a potential match. Is it possible to create a software where we could find the potential match and reduce the wastage of time? But what is the problem with creating a software? Many people find their health status to be a sensitive topic and they do not want it to be widely known. So, how do we make this process of finding a compatible match a private one? So, no need to share your ideas in the chat. This is your assignment. You have to share your ideas in a PDF format uh, within the deadline. So, I'll be sharing a Google Forms where you can submit your ideas on this. Now, before we end the session, here is a quick recap of what we covered so far and how it all relates together. So, what we saw in Arun's session, we saw how a molecular functioning, we saw the molecular functioning of cells, right? How proteins are created from DNA and RNA, and how they are built and how they function. We also learned how to detect them. Now, in this module, what we did, we discussed about different types of cells, especially stem cells, and we discussed about biomaterials and how these DNA, RNA, proteins, these biomaterials, how they all come together to form an organ. We also learned how to build an organ from scratch. So, this is called as the bottom up approach. We are starting from basic molecules and building it up to a complex organ. Here we use the laws of physics to build our understanding of biology. But this is not always possible, right? Especially in biology where the system is very complex. Let's take human for an example. The human body contains a total of 206 bones and 650 skeletal muscles, and not to mention the organ system because they are really complex. So this all takes trillions of cells, and we estimate that there are between 37.2 trillion to 200 trillion cells in our body. And each human cells approximately contains 3 billion DNA base pairs. And each cell produces so many proteins as well. Now just imagine the number of molecules that are interacting with each other every second, right? It's a very complex process. Now, what we'll be doing in the next section is the next sessions in the next week is we'll use our foundational knowledge and our expectations to interpret the things which we actually see. Now, this approach is called as the top down approach. For example, uh, you see a mouse, let's say, it is scared of you. So, what will happen? It will stay still. All we can see is the mouse staying still like a statue. But what happens inside its body? We cannot observe with eyes, right? But with our existing knowledge, we can interpret what might be happening in its body, what is responsible for this behavior of that mouse. So, how do we relate the molecules that drive these behaviors? Isn't it interesting, guys? So, we'll be covering that in the next module. And this is it for today, guys. Thank you so much for staying with me throughout this module. I hope you really enjoyed it. If you have any doubts, you can stay back, or else you are welcome to leave the session.